So we will move on to the second lecture, what we planned this morning. The second lecture is a very, very interesting topic. Genomics assisted viral diagnostics or diagnosis. This lecture is very important from many points of views. For example, the speaker is going to expose all of you to the viral genomics and how these viral genomes are quite different from other genomes. He will be giving some basic information about viral genomics, viral genome, and how this viral genome information can be used to develop highly sensitive and highly specific and cost-effective diagnostic system for plant viruses. Of course, the learning, whatever you are having from here, you can apply in other systems also because the core principles remain same. So with that, I have a pleasure in inviting Dr. Anirban Roy. He's a principal scientist in plant pathology, specialization in plant virology, an excellent teacher, excellent researcher, lot of experience in handling plant viruses, and he is one of the very constant speaker in our programs, training programs. I'm sure he will take all of you to the viral genome world, and you would be having very nice time with Dr. Anirban. Dr. Anirban, please, floor is yours.
Already afternoon. Good afternoon. As Dr. Kumar told you that for next almost 1 hour, I'm taking to the world of plant virology. Now, I'm seeing a couple of faces from these divisions. For them, it is they know about the virus. But many of you may have very little knowledge on the plant viruses. So first I'll start with what are plant viruses, what are their genome, what is the genomics of plant viruses, what are the aspects we cover in the genomics of the plant viruses, and from there I'll try to start with that how the genomics study will help us to detect viruses. Now, genomic study can help us to detect known viruses, which we already know. Genomic study also can be used to understand unknown viruses, because in the virology world, whatever knowledge we have at present, it is just the tip of the iceberg. We do not know how many different types of viruses are present. It is enormous amount because new genomics technologies like next generation sequencing are coming, and through that we are able to know that in a plant only we know about few viruses they are infecting, but besides that there are thousands and thousands of viruses are there. They may not causing any disease, just like the our gut bacteria or the endosymbiotic bacteria. They are having a huge number which do not cause any disease, but they are associated with human body or plant. Similarly, earlier our only idea is that the virus means they will only cause disease, and there is not possible that a virus is present without any causing any harmful effect. But now we are able to know that in a plant there are only few viruses which may cause disease, but a lot of other viruses do not know. The next thing is that for this genomic science enabled us to detect the plant viruses at the point of care, and that is most important part, because the problem which farmers facing or other breeders and other who are working on the plant virology or plant pathology, the major aspect is that that whatever technologies we have, if we have to use those technology to detect viruses, we need to bring the sample to our lab, and then we have to do some analysis, some the things, then only we are able to know that the virus is there. But new technologies are coming up where we do not know. We are able to actually detect the viruses without having any information, without any laboratory setup, anything, directly on the field, we can detect the viruses. And that is possible because we have different tools now available with us, and all those tools are mainly available after understanding the genome of the virus. So let us start with some information like what are viruses. It may look silly to many of you, but I know after this, when I'll click the next one, I'll ask one question, and I'd like to hear from you, not from the plant pathology students, other students. What is your idea? So you know that viruses are the small, we do not know whether we'll call it as organism or it is entity, something which is intracellular, submicroscopic, we cannot see it in a normal microscope, and they are absolutely obligate parasite. They cannot live without a living cell. And they replicate inside a living cell and produce their proteins. And for that, they don't have any protein synthesis machinery. Everything they depends upon host. So they divert the host machinery for their benefit, for their replication, for their protein synthesis. And they do only the replication behavior. They show the replication behavior inside the cell. But outside the cell, if you take it, a virus outside the cell, they do not show the property of replication. And this kind of phenomenon, many of your earlier studies, earlier days when we were in schools, a very misconception was roped into us that viruses are something different than other microorganisms. So this question often you may hear that viruses, whether they are living, they are non-living, or they are intermediate between living and non-living. So here is my question: what is your idea? Do you think that viruses are living or non-living, or they are intermediate between living and non-living? Just a few quick answer I hear from you. What is your idea? Anyone? Not plant pathology student. Intermediate. How many actually feel that they are living? Raise your hand. No. Okay. How many feel that they are non-living? No. Okay. And how many of you think that they are intermediate between living and non-living? Majority of you are thinking that viruses are is something which is in between living and non-living. Now, on what ground you are telling that thing? On what ground you are telling that viruses are intermediate between living and non-living? Anyone? Anyone? Okay. So for their replication, they have a condition. Okay. For their replication, they have a condition that they need a living host. Without a living host, they cannot replicate. So for any other living organism, do not they also behave the same? Without any condition, any organism can replicate. So if anybody is not able, and so you are considering at least one thing, that replication is one of the key point to define or distinguish between the living and non-living. Correct or not? Replication is the property of anything that can distinguish between living and non-living. This point everybody agrees? Yes? So virus replicate, but it has a condition that without living cell I will not replicate. Now tell all of you, do you think that you are living or non-living? And for viruses, why you will treat them differently? They also have a condition that with living cell, then they only replicate. Without living cell, they will not replicate. So for any organism, they have a condition for replication. The viruses also causes the same thing. They have a condition that with a living cell, they will replicate. Without living cell, they will not. So keeping this point, the virus is quite respectable as living organism, okay, because they replicate. So they can be fit in a continuous lineage. Anything which can replicate, they can fit in lineage, and that is why they are living organisms, to replicate with a condition. So that condition applies to any living organism. Without that condition, they cannot replicate. For viruses also, they replicate. Second point, their evolution quite effectively. We can trace the evolution of a virus. Okay, so any living organism find that if their evolution can be traced back. So in this point also, virus satisfied. Third point, they are independent in terms of they are not limited to in one particular organism as host. Viruses can infect any host. Even there are reports that there are many viruses infects virus. Okay, so they are not restricted to a particular. Keeping the main points which satisfy a particular entity being a living organism, virus satisfy all these. Then why we think that virus is non-living or intermediate? From after this class, should tell virus is also material. Now, next thing is that each virus has a thermal inactivity. Means if you take a virus out from a living cell, up, then after a certain temperature, virus, if apply temperature and then you try to inoculate the virus in, you'll see virus will not able to, because that time virus got killed due to the heat activity killed. Second point, every virus has a dilution end point. That means if you dilute the virus at a certain point, after that if you try to again inoculate, virus will get. Third point, longevity. Taking out the virus from a cell, keeping the virus as such in a normal condition without any living host, after certain time they will, and they will not able to revive back the life to the virus. They will not. Now the question is that which thing can be killed? If it is a living, then only that can be killed. If it is non-living, so if virus can be killed, that means, okay. So this is another proof that virus is respectable as a living. Okay. So that is why I told you that from your school days, a misconception was roped into your mind that virus is in between living and non-living, but it is not true. Completely false. Virus is living organism. Okay, definitely don't have anything. So they don't have any cellular structure, don't have any organelle, but actually they keep it simple, just like our rishi and the minimum things which is fed for. The viruses among the microorganisms, they are a different class. They are in that rishi kind of thing. That they leave everything luxury. Only they have minimum thing requirement which only helps them. Nothing else. So virus is made up of things, you know this, that virus is made of a protein cell, and within this protein cell the viral nucleic acid. The nucleic acid of the virus may be DNA or RNA, not to. Virus has a genetic material. RNA is. So plant viruses, they are of the virus, where this is, where it is a spherical cell, outer is morphological. Under electron microscopy, you will see the virus of rod separation or different emanate the different types of virus particles the virus have. Now, in this virus particle, the genome is here inside the virus particle. Now, you may have this kind of question that is the protein code is actually a kind of gabba, okay? That a gabba is there and inside a nucleic. The kind of idea students have, okay, that if hollow thing is there, a dabba and a nucleic acid is not correct, okay. So how these viruses are and the nucleic acid of the viruses and the protein they interact. Yellow color and these are unit the one layer of C entire virus particle. So it is not that there is a hollow core and then inside that the virus genome free floating. No. Okay. So these are some ideas actually I'm trying to tell, so which will help you to understand that how the virus replication, genomics, and all those things are now related with. So main criteria for classification is mainly based on composition, that whether the nucleic acid is DNA or that they are stranded or double stranded, whether they are positive or negative, and the gene arrangement in the viral genome. Things are taken into account when we classify the virus groups. Are included particles better, but primary criteria is only the genome. So as the criteria for virus classification, identification depends upon the genome of the viruses, the study of the genomics for the virus. Now, based on this genomic information, broadly group DNA viruses, RNA viruses, DNA virus has again single-stranded DNA, RNA virus, RNA. Now, when we are talking about single-stranded RNA, any RNA, if it is single-stranded, a protein can directly produce from that if the RNA is acting as RNA, or if the RNA is in the opposite polarity, the protein cannot directly. At first, it has to convert it into RNA, which can then synthesize the. So based on this standard RNA, are divided into positive RNA and negative sensing. Okay. So some of the example I'd like to tell you for single-stranded DNA viruses, virus called Gemini virus. So they have particles. It looks like the two particles, spherical particles, but it is there is only that there is no two separate particles. There is only geminate particle inside this circular virus genome. They have genes coded by this double-stranded viruses. Typical one, the genome and genes coded. Single-stranded RNA viruses are largest group of the plant virus. They have one broad and they have near genome virus or tobacco mosaic virus. Example of the negative viruses, the most world virus, the negative RNA so directly not be that, so they need a mRNA. They have three components. So there is only one genomic component genomic component, just like our chromosome, our entire genomic information is here in this is divided, their genetic information is component. All these components are one particular cell envelope, and then double-stranded RNA viruses, they have 12 different components. One particular. So these are array that how the viral looks, and all these viruses have different genes encoded with genome. Your essential genes for a virus is genes, and besides also have suppressor gene. So the suppressor the virus to overcome the fight on the host silencing machinery, which is actually trying to inhibit the virus infection. So virus also has a weapon called suppressor. Those suppressor proteins are trying to suppress the silencing effect. They may have for insect transmission. There are many other. No. So though viral genome is very small, if you compare with the bacteria, KB only, virus is 0.7, 2,700. So this is that kind of small genome available. But within that small sequence, they encode all the genes which survivability and everything. So just how clever the viruses are, they move all the cellular organs, everything, but they keep. That is why we need to study the genomics of the viruses to understand. So you know the genomic study is actually the study of any gene. Okay, virus is for the viruses. It is the only thing in the virology you have to study genome. No other morpho irrelevant for. So for the virus study means you have to study. And as for where we are actually applying the genomic study in structure, what is there in the study, that is called. So design for the viruses and the gene prediction, that what are the genes which are present in, and we can study the genome organization of the virus, how those genes are organized in all genome. This they are coming and covering structural. And comparative genomics deals with, because a lot of genome sequences of viruses are available. So when you are working with a particular, you need to first compare with the already known virus, and through that you can annotate the gene. You don't need to actually validate each gene function of the virus working, because already those things is known. Some same kind of virus, there is some sequence available. So from that sequence you can annotate your own gene. So comparison of the genome of already known viruses, the genome which you are working, called the gene. And then we can study the molecular phylogeny, the relation, all those kind of relations understand through comparative. And then functional genomics, also we can study the protein structural modeling of the how those and interaction of the viral proteins with how the host proteins and viral interact, and because to understand that of fungal pathogens, bacterial pathogens, they have certain genes in their genome which helps for their pathogenicity. Okay. They may have some enzymes, they may have some toxins, which actually is determinant cause symptoms that for the viruses have such genes, because viruses do not want to kill. So the symptom, whatever you are seeing due to the virus infection, it is not caused by the virus. It is caused by the host, because virus for their replication they need some host and they snatch it from the host. So host is now deprived of those proteins. So those proteins are involved in some pathways of the development. So when that pathway is getting inhibited, they resulted in. So that is why study of the genomics is very much important to study overall virology. It is called the diagnostic identification to understand the pathogen, how the developed, everything. If you study, we need to study the genome. So it is clear that why genomics is important for virology. Now coming to the topic of this today's lecture is that how the genomics plant virus. Now if you see the evolution of the plant virus diagnostics, then to very much relate with the development of technology. At the earlier days when there was no technology known, that time the virus were detected or identified only based on biological pathology, post electron microscopy, all those things were that time technology using which. Then when the technology developed, like the immunosorbent ELISA technique developed and other serological, then we are able to detect the viruses, the lateral. So all those technologies mainly based in which is encapsulated. Based on that, we are the virus. The biological means of the detection only detect the virus family level, because we cannot detect that, because very rough estimate what group of viruses. So when serological technique at that time we this mass of the virus is affecting, but we cannot tell which particular of the virus. So serological techniques has this limitation that much for detective events. Then different molecular techniques develop, understand the virus genomics, and that to those techniques understand the virus more precise way, and we like different formats of and so on. So now we are able to detect the virus not only at the species level, but and very next some diagnostic of which defining the AI and electronic. We can detect the viruses precisely more, and at the point mode that we can diagnostics. So what is the basic things actually how we can relate the mix with the detection, that have amplification based approach for the, and for doing that thing first we need to have the idea about the sequence of the virus, know the. So when we have the idea about the system, and we can compare the, and design primers, maybe generic or maybe specific as to genus specific virus or a species. Then we have different means for the amplification, different temperature gradient to detect the viruses, and that method formats of polyreaction, and at a single temperature also you can detect virus. Techniques are available. Combination technique or amplification techniques, the virus, and but all these techniques, the first prerequisite is information. So without the genomics, cannot design process. Now recent trends, if you see, so this situated with about the recent techniques are now very helpful, trendy now. All viruses, it is not an individual virus, detection of all the viruses, so known virus, unknown virus sequence. For that, virome analysis, that can be when we have the technology. So using the next generation, we study the entire sequence of a plant genome, which may have viral genome sequence, may have plant genome sequence, everything. Then we short out it only for the viral genome. The context we assembled and go back to the origin of the virus, able to identify what are the different viruses present that particular. It includes everything, known virus, unknown virus, and by this technology now we are able to know that in a plant they're thinking that only one, not it is viruses. So the next aspect, the point of the viruses, is a new technology called ICAR-based technology. Now it is a buzzword. Many of you heard that ICAR tool for genome editing, but that CRISPR technology can also be detect virus. In India, the very recent kit has been launched, home-based kit, Eluda, and that is based on, and in your home you can detect without any laboratory, anything. I'll talk about point a little bit more. Just I'd like to tell you about the some of you know those benefit. I'll tell something. So polymerase chain reaction. Oh, it is a artificial method of DNA using a specific that primer sequence designed based on the genomic information. Okay? So when we have a primer, that primer the template DNA which we want to amplify, that stored in standard particular temperature at 95 degrees. So when there is a then which is entered into the viral genome at a particular, then the polymerase which use as a heat polymerase, stack polymerase, they amplify that way. They amplify continuously for repeated cycles, and we are able to generate copies of the viral fragments. And if that fragment is present after gel electrophoresis, tell that virus, if not present, not there. So the next one is DNA virus. We can do like this. DNA virus, we need to add one extra. That is the RNA virus. Prime. complementary DNA sequence. Then using the amplify the entire RNA viral. In that way, using and SP a video. The basic so how we are designing the primer, and that is the genomic information. Use generic primer. So we have an example of two viruses, which is a pulse crop virus, the virus of the crop, and both of them produce like disease. So by just looking at the symptom, you cannot tell that which virus is infecting the disease. In the electron microscope or the serology test also, you cannot differentiate whether it is virus is MV or MYMIV. So that we can differentiate. But using the genomics study, MV isolates, and based on that, the genomic study, where is the sub-region present across and the IV, which the region which conserved among these, and from that, if we are able to design a primer, can amplify both the virus. So it is called a generic primer, amplify virus, and is that you can that it is MIMB infected sample or sample that can be detected, both can be detected using generic primer. We are able to detect a specific primer. For that, what we do, the same genome alignment study, and we can find out that this is the region and group, that kind of difference there. So if we use that kind of primer, only one virus will amplify, not the other. That you can only one amplify. In that way, you can specifically design a primer to amplify and specifically detect a of the virus. So this is the utility, how the genome information, the sequence, we are now converted into a primer that will be either it is a generic or a that cannot be by any other diagnostic, only by kind of or differentiate. Next thing called loop thermal. There is a requirement of a machine called ICAR machine which will convert the temperature from 5 degrees 70. So at different temperature gradient you have to give, then only the amplification artificial. So as there is a requirement of, if you overcome that dependency of that machine, so people try to find out some other DNA polymerase which can amplify DNA at a. So here is one polymerase that can amplify at a particular temperature gradient of. Here, one important thing that you need to design not primer, the prime, the designing of the prime. So you have to design this complement or portion. There is a portion where this primer we have designed that it has somewhat first primer by. Similarly, here also third primer. So how this So this method using a particular DNA polymerase which has a strand displacement activity, that is 5' to 3' polymerase, as well as the trans displacement activity, but it do not have the 3' to 5' exonuclease activity. That is why it is possible to amplify the DNA at a temperature, constant temperature, as well as it will not, because you can see that there is concatenate formation is taking place continuously. Different primers will bind and it will continuously amplify. And during the amplification, what happens, you know that DNTP molecules join at the 3' OH group, and the 2' from the 5' phosphate group, they are released as a pyrophosphate. And that pyrophosphate is deposited in the tube and makes the solution turbid. And just by looking at that thing, you can tell that whether virus is there or not. Because if the virus is there, that means it will give you a turbidity. If the virus is not there, it will not give you turbidity. Because virus is there means amplification is taking place. So you don't need to do analysis further for gel electrophoresis and other things. You can just, by seeing, you can tell the virus detection of the virus. So this is a kind of on-site detection method. Without having any instrument, you can do this amplification at a particular temperature like 6 degrees centigrade. So you need only a water bath kind of things. So you don't need any other things to amplify. So this is one of the method. But in this method also, you need to definitely isolate nucleic acid of the virus, means from the plant. So then again you need a laboratory setup to first isolate the nucleic acid. Without isolation of the nucleic acid, you do. Okay. Now next stage is to overcome. So this is the detection method that whether it is bacteria, you isolate and then you carry out this lamp amplification, and you can the result by visual detection or by gel electrophoresis, or if you have a digital chip, using that digital chip also you can detect virus. So there are different readouts of this experiment so that you can detect the virus. Now to overcome that problem, that you need an initial amplification of the nucleic acid, next technology is recombinase polymerase amplification based detection of the virus, where we do not know even we do not need to isolate the nucleic from a crude extract of the leaf. Don't need to isolate really the DNA or RNA. The crude extract of the leaf we just cross and we can able to amplify. So this the method actually it is a patented technology. Method utilizes not only the primer but as well as some proteins called recombinase protein and some protein called single stranded binding protein, and another protein is the polymerase. So what happens at the first stage, the primers they bound with the recombinase protein, and then when this recombinase bound protein comes in contact with the double stranded DNA, maybe in a very less amount, they form a recombination loop, and they cause a displacement of the one strand, and they will bind to the other strand. And when this strand is displaced, it is a single strand. Single stranded binding protein binds to this single strand. Similar things happen here also, and then the polymerase will amplify these two strands, and in that way they will finally produce the DNA, double stranded DNA. And this for this the temperature requirement is variable. You can do it at 30 to 40 temperature any time, and that can be possible that in a field condition, in a that kind of temperature, 30-40 temperature is always available. So without any instrument, without isolation of the DNA, this technology can help you to detect the virus. But one major problem for this technique is that as the amplification happens at that much low temperature, so if there is only few mismatch of the primers, that can be overcome, because at a low temperature few nucleotides if not matching, you will get a false positive. Okay, so that is a disadvantage, and that disadvantage can be overcome by other techniques which I'll tell. So this is a recombinase polymerase amplification based techniques. So just the video that these are the recombinase proteins. They will first bind to the primer sequence, and then this recombinase bound primer, they find its complement and this binding to the polymerase. So this kind of things is at a particular constant temperature, they will continuously do these steps, and from a very less amount of DNA, even from the crude sap, they will able to amplify the so this is the basic concept of recombinase polymerase. The recombinase polymerase amplification, after doing that, you can either see the result in the gel, how this is there or not. If it is there, that means virus is present. If it is, or you can use such kind of lateral flow strip, which will tell you virus is present or not. Next technique is NGS based plant virus detection, where we don't need any prior information for the genome sequence of the virus. So, but we utilize this technique is to study the genomics of the virus only. So, but utilizing this genomics technique, we can detect any viruses, whether it is a known virus, whether unknown virus, we can detect. Other technologies which I have told, like PCR or isothermal amplification, like LAMP or RPA, for that we need a known virus sequence, because from there the primer will be designed. But in this case, we don't need any known virus. What is the process? So first we have to collect a sample, and then either we have to isolate RNA or DNA, and then we have to ligate with some adapter for DNA, and after this, so we can directly do the RNA or DNA sequencing. So it will generate all the DNA sequences or RNA sequences which is present in the plant, that may have the viral sequence, that may have a plant sequence or any other sequences. Everything will come up when you do the NGS. Then after this, you have after the next thing is the data analysis. That is the most important part. There are very good softwares now available. So they will find out from this entire pool of sequences what are the bits and pieces of sequences which are matching with viral sequence. And one contig with another contig, they will do a overlap, and they will finally join the entire sequence, and they will tell that in this plant, this is the viral sequences which are present. So in that condition, we don't need any prior information of the viral sequence. Okay? So any virus sequence, that may be a non-viral sequence, it may not be non-viral sequence. So anything can be amplified, and we can able to see. So this is a genomics techniques which is used to know the sequence of the genome, and virus, everything. And using that technique, not only we are able to detect the virus, also we are able to characterize the entire genome sequence of the virus. Okay? The disadvantage of this technique, the cost is still high. Second thing is that that you again need to do the DNA isolation, RNA isolation, involvement of a sequencer, but now nanopore sequencer and all other very small sequencing machines are coming. So that made it possible now to achieve sequence information very quickly. The another important aspect for this, means negative point of for this technology, is that as it is purely sequence based and the software can do mistake. Software, if the virus may not be present, but software is finding that there is a bits and pieces of sequence that may come in the host genome also, but they will align those things, and they may tell you that this is the virus. Okay, so that virus may not be actually there. So until and unless the NGS data is supported by a biological meaningful thing, that okay, if we are able to isolate that virus, prove that whether the virus is able to replicate, may not produce symptom, but virus is able to replicate in the plant, then only we can tell, yes, it is a real sense virus. Otherwise, it is only sequence of the virus. Okay? Because during the evolutionary process, it may happen from where the viruses are evolved. They are evolved from the plant genome or whatever the host genome. From those sequences only viruses evolved. So there will be definitely some stresses of sequences are always available in the genome. If you take any viral sequence, take 10 nucleotide sequence from a virus, search it in a database of the plant genome, you'll see so many places that 10 nucleotide sequence available. That doesn't mean that that is a virus. Okay, so that is till now it is a negative point of a NGS based detection, but definitely it will help us to identify so many things without doing anything. We can characterize the virus, we can generate a lot of information about the genomics. So these are the different case studies where different viruses, unknown viruses have already detected using this technique. Now coming to the last point, which is the very recent trends for plant virus detection, is the CRISPR-Cas based detection of the viruses. So you know this CRISPR-based CRISPR technology is a buzzword, and it is linked with genome editing. Okay, so in very nutshell, if I'll tell you that this is actually prokaryotic immunity system for bacteria. Okay, so bacteria is infected by bacteriophages. To get rid of from the bacteriophages, bacteria has that kind of immunity system. In their genome, somewhere in their genome, they have a particular loci area in the genome which has a particular repeat and spacer sequence present. The repeat, so this is the CRISPR RNA, and the repeat that spacer sequence during their evolution, the spacer sequence actually copied from different bacteriophage genomes. Okay, and they have been placed into the bacterial genome. And just previous to this CRISPR loci, genes called CRISPR-associated genes or Cas genes. And some of those Cas genes are actually copying the target, placing it into the bacterial genome. Some of the Cas proteins has some other activity. So before this Cas proteins, there is another region which is called transactive CRISPR RNA loci. So they also produce a RNA sequence having a this repeat sequence. So what happens when the transcription takes place? They will produce a CRRNA and they produce a primary transcript. They are individual CRISPR RNA and they are complemented, bind with them, and they form such kind of CRISPR RNA, a duplex, a partially duplex molecule. This is able to interact with protein called Cas, which is present in this Cas gene area. So when they bind with the Cas protein, then the Cas protein configuration change, and then the Cas protein has two different nucleus motifs. One is called HNH and another one is called RUV. These two motifs come into close near close region, and then this particular CRISPR RNA directs this Cas protein to search the complementary sequence in the bacteriophage genome. So when they find that complementary sequence, the two nucleus motifs of the Cas protein cut the bacteriophage genome. That way, they got rid of the bacteriophage. So this is a bacterial immunity system. So we utilize this system for the editing in the eukaryotic genome. So what we do, instead of is to separately, we place it in one single guided RNA, which is your CRISPR RNA, rest of the portion is your tracer RNA and the loops. So this sequence we can design by ourselves based on this complementary sequence where we want to cut. So genome editing means a precise double-stranded DNA break. Okay, so when a precise double-stranded DNA break will take place, then there is methods in the plant. One is called non-homologous end joining, and another one called homology directed repair. So these two methods come into picture. So this break end, then they are joined by abruptly by putting some nucleotides or deleting some nucleotides, they are joined. So this is the property of the DNA repair mechanism of the host, and as they are joining randomly, they cause mutation in the gene and they cause abnormal termination of the proteins of that gene, and as a result, that particular protein will never form in the plant and we can get a knockout mutant. Using this homology directed repair, we have to give a complementary sequence which we need to add into that region. So if we are able to give a complementary sequence which is a flank of this, then they will be incorporated in that double-stranded break region and we can get a new edited copy of it. So in that way, we can perform the genome editing. But this CRISPR technology is not only involved in the editing of the genome, but also we can use it for the detection purpose. As I have told that this is a kit, indigenous kit made from India, the Feluda kit, which developed against the COVID-19 virus detection, which is called as the home-based diagnostic kit. Those during the COVID time. So, so that technology is now available, that how we can use this genome editing tools, that is the CRISPR tools for the diagnostics. So that is made possible because not only the Cas9 protein, now we are able to know about many other Cas proteins like Cas12a, Cas13a, and this protein, Cas12 and Cas13a, they have a collateral damage activity or collateral cleavage activity. What does it mean? That when this Cas protein, Cas12a or Cas13a, along with the CRRNA or the single guide RNA, they detect a particular sequence and they will cut that particular sequence. Along with that, they have a collateral cleavage activity in surrounding that atmosphere. If any single stranded DNA or RNA is there, they will also cut that. And as it will cut that thing, they will cause a collateral damage. So utilizing that collateral damage activity, the diagnostics has been developed. So there are two methods of diagnostics. It's called Sarlog-based diagnostics, vector-based diagnostics. So in this case, first we have to again use the RPA-based method. So to pre-amplify the viral genome, and then we have to use the Cas13 or Cas12a. So when they will bind, then they will cause this collateral damage activity of a single-stranded DNA molecule. And this single-stranded DNA molecule, if one side it is fluorophore quencher, then it will cut. The fluorophore is separated from the quencher. They will give a. Okay. So getting fluorescence means the virus was detected. If the virus is detected, collateral damage activity will take place. Then only that fluorescence will get. So you can tell that virus is detected, and this method is a real sensitive revolutionize the virus detection, because we do not need any equipment here, anything. So you just see one video. It is not the videos were working. So we are using this CRISPR based diagnostics. We are able to detect different human viruses and different viruses also we can detect using this technology. So this CRISPR based diagnostics, all different advantages and the multiplex, very short time it is taking to detect the virus, and all this whatever was the disadvantage for other methods, this method we can overcome all this. So here just I'd like to tell some of the work which we are doing in this direction, that we are working on chili leaf curl virus disease coming, but you can see below that when the plant is infected virus 3, then also you will get a similar kind of. So just by looking at the symptom, you cannot tell whether it is due to a virus infection or thrips infection or mite infection. Okay, and that actually for the proper epidemiological modeling, we need a real-time distribution of the viruses, that at that point of time how many samples are actually infected with the virus. Then we can correlate with the environmental parameters and everything. But if you do it by PCR or any other means, then you have to bring the sample, do DNA isolation in one day, then PCR in another day, then electrophoresis. So 3-4 days it will take to come up with the conclusion that at that point of time this was the virus. But by this 3-4 days time, more plants will be infected. So you cannot correlate that what is the real situation of the plant, that how many plants are infected by the virus and the environmental parameters at the real time. You cannot do this thing. But if we are able to detect on spot, yes, now at 12 o'clock, how many plants are infected by the viruses, using this technique we do, because that is a on spot detection method. So for that, we from the again genomic information of the virus, using the study of the genomics, we designed the CRISPR RNA sequence. And first we study with the plasmid DNA of the virus. We are able to detect that it is cutting the virus sequence. The uncut virus is after this CRISPR Cas12a will cut the virus, and we are able to get such kind of fluorescence based detection in the CRISPR assay. The virus, the technique is having high specificity because we are using other virus clones. It is not detecting any other virus, only the chili leaf curl virus it is detecting. But when we isolate the total DNA from the plant, try to see whether it is able to detect. It is from the plasmid DNA it is detecting, but from the total plant DNA, they are seeing that it is not detected. This indicated that this CRISPR, only the CRISPR technology is highly specific and very quick, but it is not sensitive. That means if the concentration is low from total DNA or even from crude sap, it is not able to. On the other hand, when I talk about the RPA technology, I have told you that it is very sensitive. From the total crude extract, you can detect the virus, but it is not having specificity. Okay, because at a low temperature we are doing, so it is not specific. So we are adding the RPA just prior to the CRISPR reaction. So the RPA technology we have standardized for this virus. This is from the clone plasmid. It can detect up to 3 picogram level, total DNA on picogram level, crude extract. So we don't need to isolate any nucleic acid. From the crude extract, even up to 3 times dilution, we are able to detect. So if you compare this RPA technology with the normal PCR based technology, from the total DNA, it can detect only up to 10 nanogram dilution, 10 nanogram concentration. And here this 3 femtogram concentration. So that is the sensitivity, and even in the crude sample, PCR cannot be done from the crude sample. Okay. So we standardize all different parameters, and when we study the specificity of the RPA, we observe that the RPA can detect all the viruses, okay, because it is so low temperature we are using. The specific primer which we are using for the RPA, it can, though it is having mismatches, but that mismatch will not withstand, because at low temperature everything will give you amplitude. So it is having very much sensitivity but not specific. So that is why we combine the RPA with the CRISPR, and then we come up with this kind of on-site detection that from the field plant, just using taking a lip disc, add the crude prepare the crude extract in buffer, 1 microliter of crude sap. We are taking the RPA reaction. Then after the RPA, the reaction mix of the CRISPR-Cas and the DNase alert we are adding, and we are getting the virus detection. So we can detect through total plant DNA as well as the crude. And this entire testing type is 45 minutes from crude sap in the field itself. Okay, but definitely after doing this, in this fluorescence, we again have to take that thing to the lab to see the fluorescence. Naked eye we cannot see, so we need an instrument to see the fluorescence. So there is a limitation that we have a module. We have to use a device so that we can visualize the fluorescence. Now to overcome that, we convert this technology to a lateral flow based technology so that we can visualize even itself in the field. So basic principle of this technology is that, so Cas12 gRNA, and here in this case the DNA molecule, instead of fluorescence and quencher, we are adding biotin and FAM are two we are adding with the nucleic acid, and we are using a silver gold conjugated nanoparticle conjugated FAM antibody. So when there is no virus, what happens in the lateral flow strip, we have at the test line streptavidin, which can bind with this biotin, and in the control line we have a secondary antibody to the FAM antibody. So this is the FAM antibody, the anti-antibody of that FAM. So at the when there is no virus, there is no cutting of this, so this intact molecule will go and bind here, and the AUNP FAM antibody will bind here. The excess of this AUNP antibody can also go and bind here. So as a result, we will get two bands in this lateral flow strip. But when there is a virus, so what will happen, due to the activity of this CRISPR and gRNA, they will now clip this biotin FAM into two pieces. So biotin is separated from the FAM. As a result, only the biotin will bind here. As there is no FAM, the FAM antibody will not bind here. So only all the antibody will bind to the anti-antibody line. As a result, we will detect only one band. Okay. So in that way, we can detect the virus. The absence of the band here actually tells you the presence of the virus. So we standardize all those things. We can only detect. That means they are not detected. Sensitivity, we are able to up to 7 level, the virus can be detected. Crude extract also virus can be detected. So here we don't need any instrument to take it back to see. Directly there we can see. So by using that thing, we develop this kind of kit, so which we can use directly in the field condition. And you can see that how in the field we are performing without any instrument. Take the crude sap, prepare the crude sap, then add RPA reagents, then add the UNP antibody, then CRISPR-Cas9 reporter thing, then add the UNP antibody history, and finally viruses and test also. So in that way, in the field itself, we can detect the virus without isolating DNA, doing anything. So this is the usefulness, that how everything started with the genomics of the virus, because when we have the sequence information, from there only we are able to design the cRNA region. Okay, so without the sequence information, we will not able to do these things. So here is the role of genomics, how that will help us to detect the virus. So the genomics is not only restricted to the detection of the viruses, because entire virology virus is only genome. So everything what we are studying for the viruses, everything is genomics. Whether it is so from studying viral genome, whether we need to study the virus evolution, we need to identify the viral suppressor proteins, the different proteins, their structures, the designing the inhibitors, the protein localization, everything. So susceptible, resistant, cryptome analysis, everything, whatever things you talk about, all the genomics assisted things will help us to understand the entire virology subject. Okay, so without genomics, there is no virology, because virus is only genome, so genomics is. Thank you. Yes.
Like to have you. Yeah, try to relate. Lecture. So, without have first class IP. Here.
Thank you, sir. Very much. Yeah, very good afternoon to all of you. I understand French time. Okay. Yeah, just I'll take a maximum 15 minutes time. Okay, just we may expect a couple of questions. Time is there. Okay. Hope this training you are enjoying with a lot of lectures on genomics. Okay, on microbes, insects, and pathogens. This is altogether on different aspect, very basic aspect on pathology. Okay, pathotyping. Here we are lot more to discuss on pathotyping. It's a case example I have taken as a magnum forte. Okay, what we did and how it can be applicable in other plant pathogens. Okay, so when you see what is pathotyping? Pathotyping generally we call as a characterizing the different strains or isolates of the plant pathogens based on their virulence or aggressiveness on the differential host. Okay, the host should be differential. Particular host, they should able to differentiate the pathogen nature. Okay, so set up differential host we take. Okay, for this study purpose. Very much important to understand the diversity of the pathogen so that the appropriate disease management strategies can be formulated. Why this pathotyping is important here? Okay, we see in different aspect. Okay, when we look over the pathotyping, helps for developing the resistant varieties. It helps for better disease management practices, and it helps on epidemiology aspect, track the pathogen well in advance, and to suppose any fungicide resistant strains developed or not. So to track all these things, we need a pathotyping is very important. Okay, first one we are talking about breeding for this talk much more on practical aspect, what we carried out and how the information can be shared and how it can be interpreted on other plant pathogens. Okay, so for the first one we are talking about breeding for resistant cultivars. To breed resistant varieties or resistant cultivars, we must understand the virulence or race profiling of the particular plant pathogens. Okay, so that based on the nature of the virulence or race profile, we can able to develop the then identification of the resistant sources. Once we understand the virulence or race profiling, we will end up with the pathogen categorization. We categorize the pathogen based on the race profiling. We categorize as a highly virulent one, okay, least virulent one, or medium kind of. We can have a different category. Helps to identify the virulent isolates from the first point. Then, with the help of that one, we can go for to identify the resistant sources. Okay. Then selection of diagnostic races or diagnostic pathotypes. Okay, it helps. Suppose here the differential host, what we are talking is either the resistance mechanism is known or origins are known, or it may be unknown. Suppose in case of known origins are there, in this case, this important diagnostic pathotypes which help to identify the novel resistant genes, because
we already identified the pathotypes which is already against the different origins which is already available. So based on these diagnostic races or pathotypes, we may able to identify the novel resistant genes. Then the novel resistance can be validated by already existing resistant gene based markers. That is another point. Then we can we'll come to know the knowledge on the already ongoing their varieties. What is their performance? Okay, against the dominant of the field isolates, how it is performing. So these are all the kind of different kind of information we can generate through the pathotyping. Okay, then it helps indirectly, means directly on disease management, either through the resistant varieties or through fungicides. Then it aids to develop the disease forecasting models. Okay, epidemiology, we can, by doing the pathotyping, we can able to track the pathogen. We can track the pathogen outbreaks. Okay, based on the pathotyping. Then, if any fungicide resistance happen developed particular kind of race or pathotype, that can be monitored. For all these basic studies, pathotyping is much more important. So how we carry out pathotyping? Pathotype is generally it's a process where we inoculate the different isolates collected from the hotspot location of the different places on the differential set of the host. There are different steps involved. First, selection of different hosts. Okay, there is set of differential host. I already said the differential host set able to differentiate the nature of the pathogen behaviors. Okay, suppose we are collecting certain from the different isolates collecting from the different geographical area. When you test with this differential set, it should clearly differentiate the nature of the pathogen. Okay, pathogen is based on virulence or aggressiveness, it will be able to differentiate. The differential set may be known origins. I said it is based on certain pathogens or known origins are there. For example, in case of rust pathogens, the differential set is we know very well known origins are there. Like in blast also very well known differential set. In bacterial leaf blight of rice also we have very well known differential set is there. But in certain cases, for example in fusarium, if you see fusarium infecting tomato kind of things, we have a differential set, but they are exact genetics of the resistance mechanism is not, the origin is not identified in this case. But that also serves, that differentials also serve for pathotyping purpose. It's not necessary to be a the differential set should be, it should have a known origins. Okay, even the host is differential set is having resistant or a different kind of resistance, that also serve the purpose. It's if the differential set is not available also, even with the help of pathotyping, we can able to develop the differential host set. Okay. Yeah. Then we go for inoculation. The pathogen can be challenged. Then we evaluate the pathogen. Then based on the reaction on the host, so what kind of reaction on the differential can be, we can designate as a specific pathotype. Apart from that, we have additional techniques, molecular techniques we can use, like PCR or DNA sequencing, which is using the genetic markers. Okay, we can differentiate different pathotypes. Then based on phenotypic characterization, other characters like how the colony morphology, physiological properties and biochemical, based on the biochemical test, we can able to differentiate the pathotypes. So these are all the basic steps involved in this one. So yeah, I take a case study of Magnaporthe. The story is well known. It infects different cereal crops throughout the world. Okay, in India, it is very important in rice and millets. What we did, we established the first step. I said we have to establish the isolates. So for pathotyping purpose here, because we segregate, because we in India rice we have a basmati as well as non-basmati rice. So we established from the different basmati places, starting from Uttar Pradesh, Haryana and different states. All the states are basmati rice cultivating state. Then parallelly we have taken the non-basmati growing regions also. So overall we established the this number of locations also have given. We have established around 50 isolates from the basmati and around 57 isolates non-basmati. So now isolates are there, and what next we want? We want a differential host, okay, for this pattern. So yeah, this is we face. I'll skip this one. Okay, yeah, these are all the isolates from the non-Basmati and Basmati we have established. Yeah, they come to the differential host. Have a differential host set. Okay, to see the blast this one, they more than 100 resistant genes identified in case of for the against the blast resistance, which indicates in different chromosomes. Majority of the R genes identified from the chromosome 11, 12, and 6. Okay, so are more than 100 genes. All most of the R genes are NBSS LLR region. Okay, but if you see, even though more than 100 genes identified, only around 26 R genes are cloned and characterized, well characterized, cloned and well characterized that can be used. We can use for the we can use those genes for the phenotyping purpose. So by these in this for present study we have used 25 R genes. All are the 25 R genes which is representing the differential host. The differential like this one line which is having the known R gene, okay, which is developed in LTH background. LTH is a Japonica variety. They have developed. They developed these differential lines. Each line, they phenotypically they are monogenic lines. They are to LTH except this gene. Okay, they introduced this. Suppose this line is having resistant gene. 25 include double check. It's a differential host. Similarly, we have for other pathogens also like BLB. Even we have for Rust program also. This we have inoculate. Yeah. Now we grown the seedlings. Line we grow in a propots kind of things. Once the seedling is ready, around 3 leaf stage, we takes around 15 to 21 days. Then parallelly we multiply the isolates, inoculate in the phenotyping chambers are there. The condition temperature is 25 plus or minus 1. Then humidity should be more than 90%. The light duration based on the factors. Then we inoculate this one. After 7 days, we make observation. How the particular pathogen, one isolate, how it is reacting with all these 25, 26 lines. Based on that, we observe whether the particular isolate is giving resistant to the particular line or having this own gene. Similar way, whether it is giving resistant or susceptible. Based on that, we can segregate into the pathotypes. What we did, we analyzed all the samples. Okay, we found the percent virulence of this one. We found 21 isolates categorized as a highly virulent one, 5 isolates virulent one, and 31 isolates least virulent. This is based on virulence information. Okay, this is percentage of virulence frequency of Basmati isolates, 50 isolates we have. So based on this virulence frequency, we'll come to know what all the genes are effective against those regions. Okay, because each line which is individual gene. For example, this is the resistant check, positive check. So whatever in Uttar Pradesh, all the 16 isolates tested here. Okay, all the 60 is data line is resistant to all the 60. Their virulence is 0. So similar way, all the 50 isolates we tested in monogenic lines, and we found for Basmati lines these genes are very much effective. A50, beta2, beta, and so on. So they were very much effective. Similar way we carried out for the non-Basmati also, and we found this PA12. You see, Basmati and non-Basmati, the genes are differs. Okay, but overall some kind of commonality will be there. Okay, yeah. This is now based on this pathotyping. We can deploy the genes which is very much effective for the particular region. Okay, this is in Basmati region. For example, if you take UP, these genes are very much effective. Okay, if you take in Punjab, these are all the genes are very much effective. Overall Basmati regions, PA50, beta2, beta, these genes are very much effective. Then the non-Basmati come to non-Basmati, PA12, PA9, beta2, these genes are very much effective. So based on this pathotyping, we'll able to identify what all the genes is performing apart from their pathotypes. Apart from pathotype identification, we can identify the resistance spectrum also and what all the genes are performing. Okay, overall the resistance spectrum of the genes. So overall, you see throughout the country, is PI12 performed. They show P2 is the highest level of resistance spectrum, which is shown 67%. By these genes, now it can be deployed for managing the blast disease. Okay, these genes can be introgressed into the already commercially available variety, which is maybe susceptible to the blast disease. That also can be improved based on this gene information, based on this pathotype. So we characterize the race profiling also when we found the highly virulent races overall, the isolates whatever we carried out. Whatever, this is how we did pathotyping on rice blast. This can be applicable. We can do in any pathogen of interest which you want to pursue for pathotyping. Okay. Only thing is differential host be available. Okay, with known origin or without also, even resistance mechanism also we can study. We can further can be studied. If not, we have to establish best. We have to identify the diverse genotypes, and we inoculate with the these isolates whatever we established based on the reaction of these pathogen on these host. We can develop the differential set also, but it takes time. Okay, what are the challenges we faced during the pathotyping? Okay, the first one is pathogen variability. So certain pathogens are their highly variable nature. Example in case of Magnapothe, okay, because of their clonal propagation nature, they show high level of genetic variability. Okay, then the environmental factors that can disease development, and it can complicate the phenotyping process. Okay, then this is what the key factor, the availability of the differential hosts. Okay, almost we have for the national, we have a differential for the national important plant pathogens, but still certain pathogens we doesn't have a differential. That can be, suppose any particular pathogen you are working, it has to be established. So I would like to conclude my flash talk here is that it's a fundamental tool, okay, to understand the epidemiology, develop the disease varieties, and to implement the effective practices. Okay, by accurate identification of the pathotypes, even researchers or farmers, we can devise the better management practices. Thank you very much.
Vishnu. Akani disease. Right? Calculation. Is come. Luxuriant. Anyway, so all Maya. She's funny. Like good.
Thank you, sir, for this good introduction and giving me opportunity to be here. I'm thankful to organizers. So, students, good afternoon. So, I hope you'll have some energy for next 10 to 15 minutes. I will be here. So, how many of you are aware of fusarium? As sir asked, but I could not see hand raised. Okay, okay. Many of you know what is fusarium. Huh? So, fusarium is interesting pathogen. Is filamentous. It's fungi, bacteria, or virus. It's a filamentous fungi. Contains many agronomically important plant pathogens. It produces mycotoxins also, and it's thereby opportunistic human pathogen also. There are many diseases which it is causing in human being also. So it is belongs to Ascomycetes, and it was first described by Link in the year 1809, and these are present worldwide in plants and in soil also, and its plant pathogens, they are present as endophytes also and as saprophytes also. So most members of this fusarium, they produce array of secondary metabolites. One of them, as you have discussed, gibberellic acid. It is very important. Others are also there, including some other mycotoxins also. Furthermore, this particular fusarium, it is economic. Beside this all economic importance, it serves as a key model organism for biological and evolutionary research. Okay, so that there also we are depending on this fusarium fusica. It's good model organism also. So how many chromosomes you are thinking are there in fusarium? कितने क्रोमोसोम हो सकते हैं? Any guess? So there are range of chromosomes, they are present in fusarium. Fusarium graminearium with 4 number of chromosomes, and for looking at fusarium sublunatum is 18 to 20 chromosomes. Okay, so it is having distinct genomic characteristic, this fusarium. So first, this fusarium, you know, all the why this genomic studies is important, why genome sequencing is important. So you know there are many factors influencing the style, adaptability, environment and evolution, which are not present in only this information is not available only through the coding region is not sufficient for this type of information. So you have to see non-coding region also. Okay, so for that we need to know whole organization of the genome, then only we'll be knowing what are the features, what are the genes responsible for this type of phenomenon. Further, in the Fusarium comparative genomic project, after human genome project, it was started in the year 2002 and it was completed in the year 2016. Okay, so there they sequence 3 Fusarium species. One is Fusarium graminearium, other is Fusarium oxysporum lycopersicii, and third one is Fusarium verticilliodis. Okay, so they sequenced and they submitted it. These are the bioproject numbers of this. So what they understood through this sequencing of Fusarium genome? The first thing what we came to know through the genome sequencing is this Fusarium genomes, they have remarkable genome organization. Its genome is distributed in 2 compartments. Okay, one is core and other is accessory. So core genome, it is required for normal life. Okay, so accessory chromosome, it is involved in evolution, expression, and have gene repertoire also. So they are involved in pathogenicity, and other the accessory chromosome, it is having role. They observed there are differences in organization of these accessory chromosomes also. They found in that more number of transposable elements are present compared to core chromosomes, and more host specificity is there in this accessory chromosome. So this is unique feature what they identified. Further, they identified pathogenicity chromosome in Fusarium oxysporum for my specialist lycopersicii. Okay, so they said there is one chromosome which is responsible for the pathogenicity, and this particular chromosome is mobile in nature, and if non-pathogenic Fusarium is that is transferred with this mobile chromosome, it becomes pathogenic. So there they have given some evidence of horizontal gene transfer also. Okay, and further this particular mobile, this pathogenicity related chromosome later on discovered in Fusarium oxysporum CP and Fusarium this radix cucumarium also. So, here you can see this red marked one is a mobile chromosome, and last one it is where this chromosome is absent. So they said wherever it is there, that is pathogenic. So after that, after this whole genome project, or during that time also, many these genome sequences they have come in Fusarium. So there generally in case of Fusarium, this genome size, it ranges from 33 to 65 MB, and here the Fusarium graminearium, it is having genome size of 36.4, and further this Fusarium vanitani, it is 72.9 MB. The average GC content of this Fusarium genome was almost 48.5%. There was variation in percentage of transposable elements, and it was 0.4 to 4.8%. Okay? The number of genes they predicted in these genomes, they ranged from 13,000 to 25,000. So all these information, based on this, they further in many studies they find out there is host specificity, different groups of Fusarium. Fusarium species complex, in Fusarium species complex is there, so they are much more specific to host, particular host. So apart from this, evolutionary studies, genome compartmentalization, and this pathogenicity, these genome studies, they are helpful for the identification of secondary metabolites, regulatory studies. Okay, so what are the genes? They are regulating this production of some of the secondary metabolites. Similar type of study, it was in 2013, Wayman, he studied in Fusarium fusicula. It's a good paper, you can go through it, good genome paper. So here they found that many of the Fusarium, when they sequenced 5-6 genome of Fusarium, they could find that there are many Fusarium species where all the gene involved in gibberellic acid synthesis are present. Okay, so when you see the genome, those clusters are there apart from Fusarium fusiculae in many other species also. But they observe that they are not producing that product. Okay, something is inhibiting. So here you can see like gibberellic acid, they find Fusarium fusiculae, it is in this case it is producing product, but in other case it's not there. Likewise, they identify genes involved in beta-valine synthesis and other metabolites. In many cases clusters are there, or they are not involved in the product formation. So this information also could get from genome studies. Further, in Fusarium, these genome studies, they were helpful to identify unique gene cluster which are not present in other Fusarium species. They are specific to particular Fusarium species. Like in case of Fusarium fusiculae, they identified that gene cluster PKS19 and NRPS31, it is only present in Fusarium fusiculae, okay, not in other. So this type of information, if you are getting that particular thing is only there, so how you can utilize this information, where we can utilize it? हम बताते हैं कि we are telling something is only present in that particular particular species of Fusarium. So later on you can utilize it for the development of markers and diagnostics also, because it is very unique to that particular species. This is my study where year 2017 first this Fusarium from Fusarium fusiculae genome from India. And other taking lead from that, we developed a specific marker. Transcriptome studies also we conducted. We identified some of the genes only that present in resistant genotype because of this only induced by this. We have done transcriptome of resistant and susceptible. Find some in resistant, some specific. We compared it Fusarium fusiculae isolates. We find isolate is closer to Taiwan isolate. And we also observe one cluster is unique in this Fusarium fusiculae isolate 250. Further, as I told you, we developed later on this marker we developed for real-time PCR, time-based PCR and RPA-assay also can be utilized for the resistance evaluation and seed-borne pathogen. Fusarium fusiculae is seed-borne, so for detection of the pathogen in seed and through this Fusarium genome, what we could understand where we are. This is glimpse what I have given to you. So this first this genome compartment, or it is there. It is possible if you have good sequence arranged at the chromosome level. Okay, then this contemporary genomic study have confirmed the existence of supernumerary chromosomes, that accessory chromosome what I am saying, but also have provided strong evidence horizontal transmission of the gene also. Phylogenomic allowed us to resolve many evolutionary this relationship we could establish through these genomic studies. The fusarium, there are few genomes which are assembled, best genomes that is Fusarium by King in the year 2015, by Women year 2013, and by Cooley year 2016. You can read this genome paper. These are very good paper. Ideas also for how we can proceed further. Comparative genomics, it helped develop rapid detection assays for Fusarium species. In twice, they revealed the evolutionary relationship among toxin gene clusters, whereby the loss and gain of gene the cluster may be through genomics. And now we have databases. Now we have genomes. So further, we should go for some new approaches for the comparative analysis. Nowadays, pan-genomic concept, multiple isolates, multiple strain, you can analyze through that, and you can find out the common and variation in the genome. You can go through that. Nowadays, AI-based genomic studies is also upcoming field, so you can also see that. Few databases are there specific for Fusarium. If you are working with Fusarium, you see these databases. This, I'm thankful to organizers for giving this opportunity. Thank you all.
Have lunch. Have to sign in. Have lunch and come back.
Right.